The purpose of this short mini lecture will be to review some immunological mechanisms that are associated with transfusion medicine, and that is mostly going to be focused on B cell and antibody responses and how they are generated and how they mediate tissue destruction via both intra and extravascular mechanisms. I'd remind you that the immune response is divided into both innate and adaptive components. We're going to focus a lot on the adaptive immune response and particularly B cell responses in this mini lecture. But remember that B cell responses and antibodies function in large part to go back and enhance the activity of components of the innate response. So we will also discuss the role of phagocytes, complement, and natural killer cells. Now I'm going to take you back to the lymph node, and if you'll recall the lymph node, when we talked about it in foundations, we divided it into a T cell zone and a B cell zone. Within the T cell zone, T cells are activated by MHC expressing dendritic cells that present antigens to T cells. This allows those T cells to expand to become effector T cells, T helper cells that can migrate to inflamed tissues. But some of those T cells, instead of becoming effector T cells, become follicular helper T cells. And these are the types of T cells that help B cell activation. Within the B cell zone or the B cell follicle, naive B cells are exposed to their antigens, such as a virus or a soluble protein. And for many of these B cells, this leads them to undergo minimal proliferation and form short-lived plasma cells that make IgM. But for some B cells, this allows them to internalize those antigens bound to their B cell receptor and present peptides from those antigens on their MHC class II, thus allowing them to interact with T follicular helper cells. These T follicular helper cells, in turn, can provide co-stimulatory signals to the B cells, or signal 2, and they provide cytokines to those B cells. And this, in turn, leads to the formation of the germinal center, which is characterized by massive clonal expansion, affinity maturation of the B cell response, isotype class switching. So instead of making IgM antibodies in the germinal center, often we produce IgG or IgA or IgE antibodies. The formation of long-lived plasma cells that can persist for years after exposure, and the formation of memory B cells that can respond rapidly to a second exposure. Antibody responses can therefore be very long-lived. A single exposure in the presence of T cell help can lead to the formation of both short and long-lived plasma cells, meaning you get both a short-lived IgM and a long-lived IgG response. And if you are exposed to those antigens again, memory B cells are activated, leading to the formation of more long-lived plasma cells and boosting the amount of antibody, and especially IgG antibodies, circulating in the serum. Now, there are some cases where you get persistent exposure to non-protein antigens, for example, components of the microbiota. And this can induce the continuous formation of short-lived plasma cells, even in the absence of T cell help. And therefore, you can get a long-lived IgM response present in the serum, or even some IgG through those mechanisms. So there are ways in which you can get kind of a persistent, slow-burn IgM response. There are a number of different antibody isotypes listed here. All of the antibody isotypes have a variable region which allows them to bind to antigen, and all of the antibody isotypes have a constant, or Fc region, that helps these antibodies to mediate their effector functions. But remember, each Fc region is distinct, and therefore each antibody, because they have different Fc regions, mediates distinct effector functions. IgM antibodies, for example, besides being able to neutralize pathogens, are also really good at activating complement. And these are the earliest produced antibodies. IgG antibodies, also good at neutralizing, are also good at complement activation. Additionally, IgG antibodies are good at opsonizing pathogens to enhance phagocytosis, and they can induce a process that we'll talk about in a minute called antibody-dependent cell-mediated cytotoxicity. And these are the antibodies that are most prevalent in serum. IgA antibodies are good at neutralizing, and these are present in mucosal surfaces. IgE antibodies are specialized in the activation of mast cells and eosinophils. And IgD antibodies aren't really secreted at very particularly high levels and are really only expressed on the surface of naive B cells. One other key way to understand how antibodies function is the idea of valency. And valency refers to how many antigen binding sites an antibody has. An IgG antibody has 2 antigen binding sites. And in fact, all antibodies are bivalent. The more antigen binding sites an antibody molecule has, the more this increases the overall avidity of the interaction. So IgG are much higher affinity than IgM antibodies, but IgM antibodies can make up for this a little bit because they're secreted as pentamers and therefore have 10 antigen binding sites. This can particularly help them to bind antigens that are present as repeated motifs. So what are some of the key mechanisms of destruction mediated by antibodies? I'm going to present 4 to you. 3 of them you've already learned about in Foundations. The first of those is opsonization. And this is the idea that antibodies specific for a pathogen can coat that pathogen and can be recognized by phagocytes expressing Fc gamma receptors. So these are mediated by IgG antibodies, and these are receptors that are specific for the Fc region of IgG. And this induces enhanced phagocytosis of this pathogen, which can then be destroyed in the phagolysosome. But of course, if these antibodies are specific for a red blood cell, you can imagine that you might induce phagocytosis of these red blood cells and destruction. The second mechanism is activation of complement. This can be mediated by both IgG and IgM. Complement proteins are activated by antibodies bound to the surface of pathogens. And when the complement cascade is activated by IgG or IgM antibodies, this leads to the formation of membrane attack complexes and lysis of the bacteria. But in some cases, rather than formation of the membrane attack complex, some complement fragments can bind the surface of bacteria and lead to enhanced phagocytosis. And some complement fragments are soluble and can directly act on endothelial cells and induce vasodilation and inflammation, or in other words, they are vasoactive. And all of these mechanisms can be present in transfusion reactions. Antibodies bound to the surface of red blood cells can induce complement activation and the formation of membrane attack complexes and red blood cell lysis, opsonization and phagocytosis, as well as vasodilation and inflammation in that area. The third mechanism is the allergic reaction mediated by IgE. We typically think of allergic reactions as being induced by allergens like pollen, but in some cases allergic reactions can be induced by foreign blood components. So pollens can induce the production of IgE upon a first exposure. And on a second exposure, these IgE molecules will coat the surface of a mast cell binding to that mast cell, which expresses these Fc epsilon receptors. When these IgE molecules are then triggered by exposure, a second exposure to an antigen, this induces the release of all these granules from this mast cell, which includes lots of histamines and leukotrienes, which mediate the allergic response. Again, if these are not pollens but foreign blood components, then you can imagine that you might have a transfusion reaction occur. The last mechanism that I haven't introduced to you yet is antibody-dependent cell-mediated cytotoxicity, or ADCC. And this is largely mediated by NK cells. And NK cells express lots of Fc gamma receptors, and these Fc gamma receptors can bind to antibodies to the Fc region of the IgG antibody. And in this case, let's imagine this IgG is specific for a viral glycoprotein. When viruses invade cells, many times they leave portions of that glycoprotein on the surface of cells. So this allows this NK cell to recognize this virally infected cell via its IgG antibodies and recognize the viral GP on the surface of that cell. This NK cell specializes in secreting cytolytic granules that can then kill and induce apoptosis of this target cell. Once again, if this is a red blood cell that is recognized, then these NK cells, instead of inducing killing of virally infected cells, are going to induce killing of the red blood cell. Okay, so why do antibodies attack cells rather than pathogens? Well, one key mechanism is autoimmunity, or the formation of autoantibodies. But this isn't something that we're going to talk much about today, and we will discuss this much more in host and defense. On the other hand, many antibodies are cross-reactive to commensal or microbiota antigens. An example of this are the ABO blood group antigens. Many times antibodies that induce transfusion reactions are induced through exposure to non-self antigens. This can happen during pregnancy, for example, Rh antigens. It can happen during a prior transfusion, for example, a reaction to non-self blood products. It can happen from a prior transplant, and you can induce antibody responses to both major and minor histocompatibility antigens. These antibodies that are specific for non-self MHC molecules, often we call them alloantibodies because they are induced during an allogeneic response. And they can induce a form of hyperacute rejection, which is a rapid tissue rejection that occurs within minutes to hours. Antibodies are able to mediate destruction of red blood cells through some key mechanisms. I'm going to talk about mechanisms involved with both intravascular and extravascular hemolysis. Intravascular hemolysis is an acute reaction. It's a fast rate of hemolysis. An example of this are anti-ABO antibodies, and another example can be alloantibodies that induce hyperacute rejection. Often this results in complement-mediated red blood cell lysis, neutrophil recruitment, and activation of those neutrophils, which can lead to thrombosis and vascular destruction. Extravascular hemolysis is a persistent, slower rate of hemolysis that can be induced by both IgM and IgG. This is often associated with complement-mediated opsonization, and this leads to phagocytosis of red blood cells by macrophages and granulocytes, and it can also lead to IgG-mediated antibody-dependent cell-mediated cytotoxicity mediated by NK cells. So just as an example, this is how cross-reactivity to commensal organisms can lead to transfusion reactions. And this is the example of the ABO blood groups. A and B are unique sugar patterns that are shared with commensals. But if you don't have A or B antigens, the presence of those commensals will lead to you forming antibodies, anti-A or anti-B IgM antibodies. If you do have the A and B antigens, immune tolerance mechanisms will kick in and you'll never form those antibodies. So this can lead to a severe transfusion reaction, which is an intravascular mechanism of red blood cell destruction due to complement activation. And this is very rare nowadays due to modern screening methods. An example of an extravascular mechanism of a transfusion reaction is hemolytic disease of the newborn, or erythroblastosis fetalis. This is induced by exposure of an Rh negative mother to Rh positive fetal blood during birth, or also fetal red blood cells can move across the placenta, and these can induce Rh specific IgG. And so during pregnancy with an Rh positive fetus, often a subsequent pregnancy, IgG antibodies can cross from the mother across the placenta to the fetus and induce hemolytic anemia. And again, this is an extravascular mechanism of destruction. And you can treat moms, and so good prenatal care can prevent this because you can treat moms with Rho-D immunoglobulin, which blocks the recognition of the Rh antigen. Lastly, I just want to mention that there are certain types of allergic responses that can be severe in transfusion reactions called anaphylaxis, and these can be induced by IgE antibodies. IgE mediates mast cell degranulation and release of histamines and leukotrienes, which leads to the symptoms of the allergic response. But in cases where these histamines and leukotrienes are present at really high levels, this can lead to dysregulated vasodilation, drop in blood pressure, poor organ perfusion, and eventual organ failure. And so this is a form of shock. And so if this occurs to blood products, then you can imagine anaphylaxis being induced following transfusion. Okay, in summary, immune-related transfusion reactions are usually mediated by antibodies. IgM and IgG can induce complement activation and opsonization and destroy red blood cells via both intravascular and extravascular mechanisms. IgE can induce anaphylactic responses, and antibodies may be specific for shared commensal antigens, form serum components, or major minor histocompatibility differences. The key ways that this happens is through pregnancy, prior transfusion, or prior transplant. Okay, so if you have any questions about this, please feel free to contact me. I'd be happy to address any of your questions by email. Good luck.